Good afternoon. I'm Chris Fanta. It's my pleasure to welcome you to Asthma Grand Rounds, the first for this academic year, which as best I can calculate is the 20th season of Partners Asthma Center's Asthma Grand Rounds. And we're absolutely delighted to have as our premier speaker Dr. Peggy S, who's a member of the Pulmonary and Critical Care Medicine Division at Mass General Hospital and also a research associate at the Harvard T.H. Chan School of Public Health. And she's traveled around the world to study the impact of inhaled endotoxin on the lungs. And we're absolutely delighted that you're here to talk to us about endotoxin and asthma, friend or foe. Thank you.
Thanks very much, Dr. Fanta, for the kind introduction. And it really is a great honor to be here today talking to you about the subject, endotoxin and asthma, friend or foe. I have no conflicts of interest to disclose. About a month ago, there was a paper published in the New England Journal of Medicine that was very interesting. It looked at innate immunity and asthma risk in Amish and Hutterite farm children. Now, the Amish and Hutterite are farmers who emigrated from Europe to the United States in the 1700s and 1800s. They lived in enclaves in the U.S. and they were reproductively isolated and had a very distinct way of life. In fact, their lifestyle had a lot of characteristics that have been associated with protection against asthma development, such as being in a farm environment, having a large number of siblings, not being exposed to a lot of environmental tobacco smoke, and not having a lot of indoor pets. However, epidemiologists have known for some time now that the asthma risk is quite different between these two groups of people. Recent studies have shown that the asthma prevalence in the Amish is about 5.2% and about 21.3% in the Hutterites. So these investigators wanted to figure out why. And so what they did was they recruited 30 Amish children and 30 Hutterite children, and they did very extensive phenotyping both of their immune systems, their genetic backgrounds, and their environment. One of the first things that they noticed is that the immune systems of the Amish children seem to be a little bit different. In particular, they homed in on the innate immune response. And so what they did here was they took peripheral circulating leukocytes and incubated these leukocytes with endotoxin. And they found that the cytokine production in the Amish children was much diminished compared to the Hutterites. The leukocytes really did not respond as robustly. To tease the mechanism behind this out, they asked, well, is it genetics? They genotyped several ancestry informative markers and found that there's really no evidence that the genetic ancestry between the Amish and Hutterites are different. So then, of course, the next logical question is it the environment? Well, I did not tell you that the Amish and Hutterites, although both farmers, take a very different approach to farming. The Amish tend to be very traditional, meaning they actually don't use any machines. They use horses for transportation and for farming, and they tend to live in single-family farms. On the other hand, the Hutterite families tend to work on large communal farms and use a lot of machines, as you can see in this picture. So to further figure out what the role of environment is, these investigators actually turned to a mouse model. They used a mouse model of asthma using ovalbumin as the stimulant. And what they did was they collected dust from the homes of the Amish and the Hutterite children. And then they extracted the dust into an aqueous extract, which they gave intranasally to mice from the time of birth until 7 weeks of age when they were adults. And then they did a lot of phenotyping look at asthma outcomes. So one thing they noticed is that when you look at the mice that have been treated with dust extract from the Amish homes, there was actually significantly decreased bronchial hyperreactivity compared to control mice. Whereas when you look at the mice treated with the Hutterite dust, the exact opposite was seen. There was, in fact, increased bronchial hyperreactivity. And so there was something about the house dust in the Amish homes that seems to be protective against asthma. To dig a little bit further, the investigators noted that we actually know the signaling pathways for the innate immune signaling system when endotoxin is in the environment. And so they looked at mice that were deficient in either MyD88 or TRIF, and then they put these mice to the same asthma model. And what they found was that in these deficient mice, this protective effect actually went away. So this study tells us that there's something in the Amish house dust that is protective against asthma, and that this happens through innate immune signaling. Now, what is this magical compound? Can we bottle it up and give it to all our children and prevent them from getting asthma? Well, the investigators looked at endotoxin levels in the Amish versus the Hutterite homes, and they found that on average, endotoxin levels were much higher in the Amish homes. And so, do we have this solved? Is endotoxin a friend of asthma? Well, if you take this study, which is very compelling, and you take it in the context of the literature, you find that the literature is actually extremely confusing. And so, for example, this is by no means an exhaustive list, but there are as many studies saying that endotoxin exposure is protective against asthma development or asthma symptoms, as there are studies saying that no, actually, if you're exposed to endotoxin, it's actually harmful. You could argue with me that, okay, well, these are studies that are heterogeneous. For example, some are cross-sectional, others are longitudinal. Maybe they measured endotoxin in different ways at different times, or they adjusted for different covariates. But let me point you to this one study. So this is not a typo. This is the same study, and I listed it in each of the same 3 categories, because what these investigators did was they took 3 large European birth cohorts who had endotoxin measured in house dust from the first year of life, and then they looked at asthma outcomes in a very uniform way from the ages of 6 to 10. What they found was that in the Spanish cohort, if you're exposed to endotoxin in the first year of life, you were actually protected against ever developing asthma by the age of 10. In the German cohort, there was no association whatsoever. And in their Dutch cohort, actually, endotoxin exposure was associated with an increased risk of having active asthma by the age of 6. And so the authors of the study concluded that the results of our analysis are inconsistent. Both positive and negative associations were observed. And I think that really pretty much sums up the literature. It's extremely confusing. We don't really know. I thought today what we would do is actually go through a few things that I think might help tease out why we see these inconsistencies. I wanted to talk about how the hygiene hypothesis has evolved, because this is the basis for why we think microbial exposure could be protective. And then I wanted to talk about how we measure microbes, both in the studies we have been doing and possibilities. And then I want to home in on a few studies looking at microbes and asthma, and finally talk to you about a few special cases. So the hygiene hypothesis was first proposed by Dr. Strachan in 1989. What he noticed was that in large epidemiologic studies, if you have more siblings or if you come later in the birth order, you actually are protected against developing allergy. And so here what I've depicted is data from 2 large UK birth cohorts from 1958 and 1970. And on the x-axis, what you can see is birth order. So 2 means you have 1 older sibling, 3 means you have 2 older siblings, etc. And on the y-axis is the prevalence of hay fever diagnosis by the age of 16. So you can observe 2 things from this data. The first is that the later cohort from 1970 seems to have a much higher prevalence of atopy. And second, that if you come later in the birth order, there seems to be a dose response, a protective effect of being later in the birth order and getting hay fever. To explain this observation, Dr. Strachan came up with this hygiene hypothesis, where he said if you have more siblings, you're probably more likely to get infections early in life, and that is protective against atopy later in life. Others have come later and said, well, this is explained by this Th1 to Th2 tension. If you get a lot of infections, you're more Th1 prone, and so you don't get Th2 diseases like allergy and atopy. Let me take a quick moment there to point out something that Dr. Strachan knew as well, which is that atopy and asthma are not the same thing. And so on the right now, I've depicted the data for asthma as an outcome instead of atopy, and you can see that there's no clear association between birth order and asthma. And so when we read the literature, we must distinguish between atopy and asthma because they are not the same outcome. So this was the hygiene hypothesis in 1989. In the decades that followed, we tried very hard to figure out whether this was true or not, and there were a few things that were inconsistent. So first of all, there's actually no clear evidence that early life infections is actually protective against allergy. And so even Dr. Strachan himself published a paper later saying that this was not the case. Simultaneously, investigators around the world also noticed that in developed countries, it's not just Th2 type diseases like allergy or asthma that were increasing, that in fact Th1 type diseases like type 1 diabetes and inflammatory bowel disease and autoimmune diseases were also increasing. And so our theory actually doesn't hold up to the observations that have come after them. In 1998, Dr. Wold actually published a long editorial where he talked about the importance of the gut microbiota. And so he made a few observations. The first was he'd done studies actually in the gut microflora of Pakistani versus Swedish infants. And he found that Swedish infants actually acquired gut enterobacteria much later in life, and when they did, it was only a single strain. In contrast, Pakistani infants actually have a succession of multiple different strains of enterobacteria. This, combined with the observation that in a mouse model, if you give an oral antigen and they react to that antigen, you can abolish that reaction, that is to increase tolerance by giving it with a microbial product like endotoxin or other microbes. And so he hypothesized that in the age of increasing hygiene, there's actually decreased transmission of bacteria by the fecal oral routes. And that decreases the amount or the types of gut enterobacteria, leading us to be less tolerant to antigens and leading to increased atopy. In 2004, Dr. Rook came up with also a similar hypothesis. And so his observations were centered around germ-free rats and mice who seem to be quite sickly. They have diseases like arthritis and colitis and things like that. But if you reconstitute their gut flora with normal commensal flora, many of these symptoms are actually abolished. And so his thought was that the issue was not pathogenic bacteria, it's all these commensal bacteria, which he called old friends, that are important for educating our T regulatory cells and when to react and when not to react. And without this education, there was an increased incidence of allergic and autoimmune diseases. Finally, Dr. Blazer in 2009 also came up with a related hypothesis. And so his concern was that it seemed like microbiota in our bodies are actually disappearing across generations. And so he looked at H. pylori as an example. When you look at NHANES data and you look at people born in 1915 or earlier, the prevalence of H. pylori antibodies was about 60%. But if you were born in 1995 or later, that number is less than 5%, with a concern that it will soon be 0%. And he was worried that once we lose these microbiota, we can't ever gain them back again. And so with these increasing hygienic lifestyles, we're actually having a generational loss of ancient human microbiota, and that decreases the opportunities for immune education, increasing first world diseases. So the common theme here, I think, really is that the microbes do play a very important role in educating our immune system, but we don't really understand which microbes are helpful and how that actually plays out. I wanted to tell you about the study that was just published this year that actually really describes in great detail how this is possible. And so I love the study because it's also rooted in an epidemiologic observation, but then it goes back to the bench. So this study is focused in Karelia, which is part of Northern Europe that has a very distinct culture. However, because of generations of sort of geopolitical conflicts, this region is actually split between several different countries. And so you have Finland, which is very wealthy and very hygienic, and then you have Russia, which is less wealthy. And it's been known for a long time that there's actually a gradient of autoimmune and allergic diseases across these countries, being highest in Finland and lowest in Russia. And so these investigators wanted to look further into the role of microbes in this epidemiologic phenomenon. So what they did was they recruited actually birth cohorts of infants in each of these 3 locations, and they collected stool every month. And they were interested in type 1 diabetes, so they also tested the sera of these infants for autoantibodies for type 1 diabetes. And so this is what they found. So when you look at the gut microbiota, what you can see is that the Finnish infants have a different gut microbiota. It's predominated by Bacteroides, whereas the Russian infants seem to have a lot more Bifidobacterium. But what really caught these investigators' attention was this bacteria called Bacteroides dorii, which seems to be associated with type 1 diabetes in other studies. And so most studies would actually stop here and say, interesting observation, I wonder what. Well, they took this even further. They actually took all the different species of bacteria that they isolated from the infant guts, and they actually isolated the endotoxin. And they looked at it, and the molecular characteristics of the endotoxin was actually different. The endotoxin was different structurally. They then said, okay, well, the endotoxin looks different. Does it behave differently when you expose it to human cells? And so what they did was they took human peripheral blood mononuclear cells, or human PBMCs, and they incubated the PBMCs with the same doses of each type of endotoxin. And what you can see is that there's actually a lot of variability. For example, if you looked at Bacteroides dorii, it actually elicits no cytokine response whatsoever from the human PBMCs. On the other hand, with E. coli, you can see that it elicits a very robust immune response. Now, it's actually not novel. People do know that different endotoxin from different species actually can elicit different responses, but what they did next was actually much more interesting, which is that they started to mix endotoxin from different bacterial species to see what happened. And what they noticed was that if you have a 10-to-1 ratio of Bacteroides dorii to E. coli, that the ability of the E. coli endotoxin to elicit a response was actually completely abolished. And so if you believe that, being able to elicit an immune response is part of immune education, then this is actually a very important observation. To take it back to sort of a disease model, they took actually a type 1 diabetes mouse model, and they injected either Bacteroides durii, E. coli endotoxin, or a control. And they found that the mice treated with E. coli endotoxin actually did not develop type 1 diabetes, or when they did, it was much later in onset, whereas those injected with Bacteroides durii did not have the same protective effect. And so this is a very elaborate story to point out that not all endotoxin is the same, and that in fact, the way endotoxin from different species interacts with our immune systems to teach us to develop disease or not is actually very different. And I think this begins to tell us why our studies are so confusing, is that endotoxin just isn't the same. So if this is the case, then let's ask, how are we actually measuring endotoxin in our studies? Well, the most common assay used is something called the Limulus Amebocyte Lysate, LAL assay, which tends to measure potency, and it's quite a mouthful, so let's break it down. So Limulus stands for Limulus polyphemus, which is actually the scientific name for the Atlantic horseshoe crab. Amebocyte stands for blood, and lysate refers to the contents of the blood. And so what does horseshoe crab blood have anything to do with endotoxin? I think this is a really cool story. And so in the 1950s, a marine biologist at Woods Hole, Massachusetts, was working with horseshoe crabs. And he discovered that one of his crabs had been infected with gram-negative bacteria. But the interesting observation was that, in fact, all the blood in this crab had coagulated. And so he went on to do a few other studies demonstrating that if you take gram-negative bacteria, whether dead or alive, on contact with horseshoe crab blood, it causes it to clot. And so this becomes the basis for our assays. The amount of clotting tells us how much endotoxin is in the system. It would be remiss of me not to mention, though, that this system is actually not specific to endotoxin, that in fact, fungi can also activate this. 1,3-beta-glucan is present in fungal bacteria, fungal cell walls, and that can also activate this clotting cascade. So many of the assays we use commercially actually have something to try and block this activation of factor G, to try and make the assay more specific to endotoxin, but truthfully, it's not 100% specific. So this is how we measure endotoxin, and when we take a step back and think about it, our choice to use this assay in our epidemiologic studies suggests that we think that what happens in horseshoe crab happens in people as well. And I think this is a big leap because studies have shown that that's not true. So if you take something that contains endotoxin, you measure it with LAL, and then you compare it to incubating endotoxin with a human cell, the reactions are very different. There's not a great correlation. And so I think this is also another reason why our studies are confusing, is that I think our LAL assay, which we use in almost all of our study, doesn't actually capture the human response to endotoxin from different bacteria. So if LAL is not the answer, then what are our other options? Well, there are several different ways we can use to detect microbes. So one way is to focus on what is alive and culturable. And this is sort of the most traditional way. So we can use microscopy in culture to try and figure out what grows and identify it with staining. An issue with this method is that it's actually not very sensitive. And so some bacteria are just very finicky. They just won't grow. And in fact, a few studies have demonstrated that probably less than 1% of what is present in your environmental sample can actually be grown. And so this method is not used very often in epidemiologic studies. Another way to think about it is, well, how much bacteria, how much fungus is there? And we tend to focus on fragments of the bacterial or fungal cell wall. So for example, endotoxin for gram-negative bacteria, or peptidoglycan for gram-positive bacteria, or the glucans for fungi. Now this is not the only way to say how much, because there is another method called quantitative PCR, where you can take a gene that you know is present in, say, every bacteria and actually use qPCR to count the number of DNA copies. And so that is another way. Finally, there's been a lot of interest in figuring out who is there, that is, which microbes are actually present. And this has been possible because in the last decade, next-generation sequencing technology has really been advanced. And so there's two main approaches to sequencing. So you can do amplicon sequencing, and by that I mean you take a marker gene. So as an example for bacteria, a marker gene is the 16S ribosomal RNA gene. It is present in every bacteria, but it has enough variation so you can use it as a fingerprint. So you have a primer that amplifies the marker gene, and then you sequence and figure out what's in your sample. Another approach is to do shotgun sequencing. And so in this case, you're not focused on one gene or gene fragment. You're actually focused on all the genes present in your sample. And so the advantages of shotgun sequencing is that first you get a much higher level of resolution. You can actually identify bacteria down to the strain level, whereas amplicon sequencing usually is you're restrained to the genus level. Another thing that shotgun sequencing can do is that besides telling you who is there, it can tell you what they're doing because you're sequencing now all the genes, and so you know the metabolic potential of your sample. Shotgun sequencing is not easy because you need a lot more DNA to start off with, and also there's a lot of human DNA that often contaminates your sample, and it's much more expensive, but that is an alternative. So a lot of investigators have said, okay, I haven't done sequencing, but I've measured endotoxin. And so if I have a lot of endotoxin, that implies that I have a lot of different types of bacteria. And the question is that actually true? And so we were curious too. And so we undertook to ask this question in the setting of animal care workers. And so we chose animal care workers because they have been known to have a high incidence of occupational asthma. And that's been associated with microbial exposures at work. And so what we did was we chose 4 different locations in 4 different animal facilities. And so let's take a little tour through where we went. And so the animal room is where animals are housed in between experiments. And the cage wash area is where after the animals have been sacrificed, so you're left with these dirty cages that have to be cleaned and processed. And so in the dirty area, you take these cages, which are filled with used litter and half-eaten food and mouse feces, and you have to dump it out into these big trash cans. And you throw it into a big washing machine. It comes out on the other side in the middle area where these wash cages are set in racks to dry. And finally, in the clean area, then these dried, clean cages are refilled with litter and refilled with feed and then autoclaved. So what we did was we set up these air samplers for 8 hours during the typical workday, and we had paired sterile filters. And for the first filter, we sent it for endotoxin and mouse allergen analysis. And the second one, we did quantitative PCR to count how many bacterial copies are there. We also did sequencing to figure out who was there. When we looked at our results, first focusing on endotoxin, you can see that the levels are actually extremely low. These were very well managed facilities. The second thing to note is that there actually are no significant differences in endotoxin level between areas. However, when you look at the 16S copy number, what you can see is that it's much highest in the dirty area, which makes sense because this is where the dirtiest work is also happening. Mouse allergen levels are also the highest in these areas. When we compared endotoxin to 16S quantitative PCR, we found that the correlation was very modest, only 0.62. And so it's difficult to say that because you measure endotoxin, that you actually know how much total bacteria is there. Now let's look at the sequencing results. And so this depicted here on the x-axis is the relative abundance of each bacteria. On the y-axis is each location. And so each color signifies a certain bacterial phylum, so looking at a very high-level view of the bacteria present. So you can see, for example, that there is a lot of green in the animal and dirty areas, and green corresponds to the firmicutes, which makes sense because when we looked at the bacteria, it was actually a lot of mammalian gut bacteria, whereas the middle area is predominated by proteobacteria, and the setup area is predominated by cyanobacteria. Now, remember, endotoxin levels are actually really not that different between these locations. And so the question is, can you really infer by an endotoxin measurement how much of what type of bacteria is actually present? Well, to do this statistically, we can look at, for example, the richness of the sample, that is how many unique taxa are present in each sample, and correlate it with endotoxin levels. And the correlation was quite modest, 0.47. And so I don't think endotoxin is a great surrogate for either bacterial diversity or community composition. I think this is another reason why our studies are confusing, is that it's not quite capturing what is most relevant to disease. So if this is the case, then where do we go? Well, fortunately, the last year has been kind of a blockbuster year for studies looking at microbial identity with sequencing and asthma. So let's review a few of these studies. The first study centered around the question, does early life microbial exposure instead of endotoxin exposure impact asthma development? And it's based on the Eureka birth cohort. And so this is a birth cohort based on 4 different US cities. And what they did was they followed babies from birth until the age of 3. And so every year they collected living room dust and they looked at allergen levels in the dust. Then they looked at clinical outcomes at the age of 3, so things like atopy based on skin testing and IgE testing, and also recurrent wheeze as a surrogate for asthma. The first thing that these investigators noted was they're kind of surprised because if you're actually exposed to cat, mouse, and cockroach allergen, you're actually less likely to develop recurrent wheeze. And this is very surprising because it runs counter to this dogma. Would we give allergens to our kids to prevent asthma? They also noted that this is a very time-specific thing, meaning that it was the exposure in the first year of life that seemed to be the most protective, whereas in year 2 and 3, the association was really not so robust. And so taking a step back, they're like, this is really very confusing. Is it possible that allergen exposure is simply a marker for microbial exposure? If you have a mouse in your house, these mice have microbes, and so maybe you just have more microbes. The investigators went back to their Eureka cohort and they did a nested case control study. And so what they did was they took their toddlers and divided them into 4 clinical groups based on whether or not they were atopic and also whether or not they were wheezing. Then they went back to the living room dust samples from 3 months of age and they looked at bacterial composition. They didn't use sequencing, they used screening microarray, which is a cheaper way to look at how many different bacterial taxa are present in your sample. And so this is what they found. So in this figure on the y-axis is microbial richness, that is the number of unique taxa present in each sample. On the y-axis is each different category. So the neither group is the group of infants that are the healthiest, that is they had neither wheeze nor atopy, whereas both means they had both wheeze or atopy. And they found that the neither group actually had the most diverse microbial exposures early in life compared to those that had both. Now, I mentioned that there was allergen exposure and there was also microbial exposure. So to look at it in a more comprehensive way, they divided the exposure into 4 different microbial categories based on microbial richness and allergen levels. And so purple would mean low microbial exposure and low allergen, and red would be high microbial and high allergen. This is the figure that they present, which I find it to be a little overwhelming, but let's just focus on the purple and the red. And so you can see that those healthy infants who had neither wheeze nor atopy also were most likely to have been exposed to high levels of microbes and high levels of allergens. In contrast, the infants that were wheezing and also had atopy were least likely to be exposed to high levels of microbes and high levels of allergens. And so it seems that there's a combination of microbial and allergen exposure that seems to be protective. And why would that be? Well, some have theorized that we know that when you give a microbial exposure with an allergen, that actually increases tolerance. So maybe that's the mechanism. Others have thought that perhaps the allergen exposure is still just a marker for microbial exposure, so it's not quite settled. Now, the idea that a wide diversity of microbial exposure is protective against asthma also has been demonstrated in a prior study from 2011. So this was actually a cross-sectional study trying to explain why a farming environment might be protective for asthma. So they looked at a lot of children in a farming and non-farming environment. And they didn't use sequencing, they used actually culture and a method called SSCP, which also used DNA variation to look at the type of bacteria present. And so this is what they found. So let's focus on the blue lines first. So if you were living on a farm, you're more likely to be exposed to a wide number of different bacterial taxa and also to a wide number of different fungal taxa. Looking at asthma, if you're exposed to a large number of bacterial taxa, you are least likely to have asthma, and that holds true as well for fungi. And so I think the literature to date really does suggest that diverse microbial exposures very early in life seems to be protective against asthma development. What happens though if you already have asthma? Does the same relationship still hold? Well, again, like I said, this is a blockbuster year for microbial exposures and asthma, and so this study was just published a few months ago. And so in this study, investigators took a cohort of school-aged children with active asthma, and then they also collected living room dust. And they also used a nested case control study. So they looked at their children with asthma and took the extremes, the mild versus severe asthma, based on medication use and symptoms. And they chose 200 of these children, and they did very extensive phenotyping, actually, of the dust that was collected. And so they did 16S and ITS sequencing to look at the richness of bacteria and fungi present, and also they divided the fungi into allergenic versus not. Then they looked at quantity by using quantitative PCR, and also quantitative using endotoxin and 1,3-beta-D-glucan. So this is what they found. For the bacterial exposures, they found that if you were exposed to low bacterial richness, that is not a lot of different bacterial species, that was protective against severe asthma. Or to put it in a different way, if you're exposed to a wide diversity of bacteria, when you have asthma, that actually was associated with increased asthma severity. Now, note that this is actually in direct contradiction to what we saw with children and protection against asthma. We saw that in birth cohorts, if you're exposed to a lot of different bacteria, that's actually protected. I do also want to highlight that measuring bacterial quantity using qPCR and endotoxin also gives very different results. The effect estimate for qPCR is about 1 versus 2.46 for endotoxin. When you look at fungi, what they found was that if you're exposed to a lot of allergenic species, or if you're exposed to just a lot of fungi, period, that was actually associated with worse asthma. And again, looking at the two ways of measuring quantity, fungal concentration with qPCR and 1,3-beta-D-glucan, which measures the cell wall component, you can see that the effect estimates are in complete different directions. So to summarize, diverse microbial exposures after asthma has been established may actually be harmful, and that fungi actually do matter. So let me take a few minutes and just talk about a few special cases. I want to convince you that at very high doses, actually, that endotoxin can act as a very nonspecific pulmonary irritant. And so this is a human study that was done in 1989. Dr. Relander and his colleagues took Enterobacter glomerans, which is the major gram-negative bacteria in cotton dust. He extracted endotoxin, and he also homogenized the bacteria. And then he recruited 77 healthy adults who had neither asthma nor atopy, and actually had them inhale concentrations, higher doses of these things. And so what he found was that at the highest doses, at 300 micrograms, half had airway irritation, 44% experienced chest tightness, and on average, they lost about 6.6% of their FEV1. So this is just to show that these healthy adults without atopy or asthma actually have asthma-like symptoms at very high doses. I think this really matters because dose is often highest in the occupational setting. And so what I have listed here is a few studies looking at airborne endotoxin levels. And I've listed the maximum endotoxin level that they measured in their study. So for example, in urban homes, it's about 20 endotoxin units per meters cubed. In urban schools, which one could argue is an occupational environment for kids, it's 780. And then in an agriculture setting, so I'm talking about large livestock farms with thousands of pigs and cattle, it's about 10,000. So I think occupational settings may be a special case because your exposure level is so high that even if you don't have asthma, you have asthma-like symptoms. And so these studies need to be parsed in a little bit different way. I'm going to return to the study I started with, the Amish Hutterite study. And so this is the main figure they had on exposure in their paper, looking at endotoxin levels between Amish and Hutterite homes. Now, buried in their appendix is actually this other figure, which I think is where the story lies. These investigators actually did not have enough dust to do sequencing in every single sample. So at the end of their study, they pulled all the dusts from the Amish homes and all the dust from the Hutterite homes, and then they did 16S sequencing. And this is what they found. And so these pie charts represent the distribution of different families of bacteria. So one color means one family. And so you can see that the composition of bacteria between the Amish and Hutterite homes is actually very different. And that in fact, if you focus on the Lysate family, that 35% of the bacteria from the Amish homes actually come from this family, whereas none actually in the Hutterite homes come from this family. And so I think, again, this illustrates the paradox is that we've been measuring microbial exposures using endotoxin, but perhaps there's a lot more of the story buried in who is actually there. So let me give you a few concluding thoughts. First, I do believe that endotoxin can be both a friend and a foe. It really depends on which bacteria it's coming from. It also depends on when you're exposed. I think one limitation is that current assays for endotoxin are simply not a good surrogate for bacterial community composition, and that we will learn a lot more in the next few years about how microbial exposures affect asthma by looking at results of sequencing. Finally, non-bacterial microbes like fungi and probably viruses and parasites also do matter. Thanks so much for your time. I'll be happy to take any questions.
What a great presentation. Thank you so much. We're open for questions. I had the sense that there are about to launch early trials of introducing microbial products or microbes to children at a young age. And would you make a guess as to when, if you were to organize such a trial, at what age should these organisms be introduced? Are they inhaled or swallowed? And do you have a favorite organism or group of organisms that you would put into the cocktail?
This is a great question. So I know that Fernando Martinez, for example, has a large RCT about to launch. And so the trial is giving infants something called bronchovaxum, which is a cocktail of pathogenic bacteria that have been killed, essentially. So you're giving killed bacterial products to children. And in preliminary trials, it seems to protect against asthma. And so the question is, will that also, allergy. So the question is, will that also be protective against asthma? I think these microbes weren't chose based on environmental sequencing studies because they actually haven't happened. And so I don't really know. I mean, there's been a lot of interest actually in lactobacillus and its role actually in being protective against asthma and things like that, but I think we just don't know. And so we're gonna learn a lot more. I think with the bronchovaxum trial, I think one interesting thing to think about is that a lot of studies have shown that in the indoor urban environment, the microbes in there are actually from the people in there. And so is it possible that these associations we found in the environment are actually reflective of the people and not actually of the environment affecting the people? I don't know. And so I don't have a great answer. I wish I did, but I think we'll know a lot more in a few years about which microbes are good, which microbes are bad, or is it just you need a certain community, or is it that it's not about the microbes but about what they do, so the metabolites they produce?
And it's all focused on gut microbiota, not respiratory tract microbiota, is that right?
Yeah, I mean, most of the studies have focused on gut microbiota because it's easy. So for example, it's easy to collect stool. It's much harder to take infants and do a bronchoscopy on them. We do, with a colleague actually at M.P.H, we do have a randomized control trial where we're introducing chickens actually to women living in rural Uganda, and we're looking very extensively at their airway and stool microbiome to see if that changes in this RCT, and also whether their lung function, lung inflammation, and various systemic inflammatory parameters do change. And so that could be interesting as well. But I don't know, is it the respiratory microbiome or the gut microbiome that matters? We don't really know, or both.
Very nice talk, thanks, Peggy. One interesting thing about, is it, you get the same response from the respiratory system than the gut, is that I believe that in the mouse model of the Hutterites versus Amish, they introduced the microbes into the lung. And in the mouse model that Sue Lynch was involved in with the dust from the dog households, they gavaged the mice. Do you think that, what would be your preferred experiment with mice and house dust?
Can you
Or do
repeat
you think
the thought?
Yeah.
So,
Yeah, so what Diane mentioned was that in the Hutterite Amish mouse model, what they did was they gave intranasal microbes, whereas Susan Lynch has done a lot of studies, and most notably there was a great PNS paper looking at introducing microbes by gavage into the gut of the mice and how that protected against asthma. I don't really know, because when you give mice microbes, intranasal just means putting things on their nose, so it's possible that they could also ingest it as well. And so I don't really know what's the best way. I mean, I suppose you could anesthetize the mice every so often and then introduce microbes that way, and that would really be able to tell you sort of which way is best. I think it's a little challenging because you don't want to infect the mice also and give them pneumonia, so I don't really know what's the best way.
Thanks for that talk. Very interesting. Another area besides the human microbiome that is overlaps with this is the building microbiome. And there's been a number of TED Talks by a woman named Jessica Green following very carefully buildings that have had their microbiology very carefully measured. If it's a naturally ventilated space, it reflects the outdoors, and if it's a mechanically ventilated space, by and large, it reflects people in it. But it's a field that is looking for a connection to human health or well-being, and obviously for office buildings and things, doesn't have much to do with early exposure. Probably for apartment buildings, it might have to do with early exposure with regard to asthma. But it is the same methodology of 16S being used a lot to describe the building microbiome that we're all subjected to.
Yeah, no, that's definitely a great point. There's a lot of interest actually in the built environment and the microbiome within. I mean, not just for asthma, actually. There's a large hospital microbiome study actually going on, looking at what happens when you take a brand new hospital and you put patients in there. Is the microbiome reflective of the other people or of the hospital? And from what I heard, actually preliminarily, if you're an inpatient, the microbes you get colonized by the building, whereas the opposite is true in a residential home. You colonize the building, and so there's so much to be learned. It's a very exciting time to be in research right now.
Yeah, just one quick question. As practitioners, we tell our patients, 4 or 5 cats would be all right, but only up to about age 1. And what's your guess about pets in the home environment?
Okay, you're asking me to speculate. I don't know. I mean, there have been a number of studies saying that cats are bad and dogs are good, but then there's also been studies saying that's not actually true. I mean, I think for the practitioners, maybe the important point is to not sweat the dirt, and that, don't, use antibacterial, hand soap and things like that on your children. It's probably not beneficial and probably might be harmful. We just don't know. And so I think the emphasis really is on don't worry about getting too clean. It's probably not too good for you.
Sounds like a good place to stop the discussion. Eat dirt, don't bathe too often. Great.
Okay.
Thank you very much for a terrific presentation.